in regulating gene expression under physiological and pathological conditions. So over to you, Dr. Shan.
We have a laser point.
Sorry, pointer. Do you have?
No point. Okay. Okay, so let's get started. Good morning, everyone. First of all, I would like to thank the organizers for the invitation. I'm very pleased to be able to present our work to the colleagues in this conference. So my name is Ming Chen from Hong Kong. So our group has three major research directions. The first one is to decipher the mechanisms of mutant H2 histone H2B in cancer development. And we also have other research directions, such as to identify novel protein factors and elucidate the mechanisms in the X chromosome reactivation, which is also a very classic example of epigenetic gene silencing. And we are also currently developing a platform to perform in vivo CRISPR screening in living animals. Before I go into the details, I would like to give you some very brief background about the histones and the chromatin. As we all know that our genomic DNA is packaged in the form of chromatin and stored in the nucleus. There are two major types of the chromatin. One is called the heterochromatin, the other one is called the euchromatin. As you can see from the schematic, the heterochromatin is a more condensed structure. So genes located within the heterochromatin usually are silenced, are not expressing. While compared to the heterochromatin, the euchromatin has a less condensed structure, and genes located in the euchromatin is usually transcriptionally active. This so-called beads on a string structure, the bead is called a nucleosome. And in each nucleosome, there are 4 copies of the histones, the core histones. They are the histone H2A, histone H2B, histone H3, and histone H4. And in each octamer, and in each nucleosome, there are a total of 147 base pair of the double-strand DNA. So 147 base pair of DNA plus the 2 copies of these 4 core histones form one single nucleosome. And you can see that there are the different red lines. There are the histone N-terminal tails, which protrude away from the core nucleosome. They are very important for the histone post-translational modifications, which I will tell you more about these in the following slides. So the DNA is packaged in the form of chromatin and stored in the nucleus. And the histones are important proteins, which are responsible for the packaging of the double-strand DNA, condensation of the DNA to store in the nucleus. If you look at these crystal structure of a human nucleosome, you can see that the nucleosome is this shape with this shape histone core, 2A, 2B, 3, and 4. And the double-strand DNA is tightly wrapped around 1.7 turns, around 147 base pair, to make one single nucleosome. So to tell you more about the histone modifications, I would like to make the story simple. So here I only show you the N-terminal tails of the histone H3 and histone H4. And here I only highlight the key lysine residues, which can be modified by the different enzymes. For example, you can see here that these are the histone H3, lysine 4, lysine 9, 27, and 36. They can be modified. For example, the lysine 4 can be methylated by the Z1A1B or the MLL complex. And the methylation of the lysine 4 of the histone X3 are the active marks. K4, for example, K4 trimethylation is an active mark which can be present in the promoters of the actively transcribing genes. While on the same histone N-terminal tail, lysine 27 of the histone X3 can also be methylated. And the trimethylation of the lysine 27 of the histone X3 is a repetitive mark, which are genes that are epigenetically silenced, are usually marked with these histone modification at the promoter region. So in the past decades, many of these enzymes have been found mutated in different cancers. For example, the MLL complex have been found mutated with gene translocation in different leukemias. And the EZH2, EZH1, EZH2 enzymes have been found mutated in prostate cancers. So it has been known that these enzymes are mutated in different cancers because they affect the histone modifications. For example, the EZH2 activation mutation found in cancer will lead to constitutive active activity of the enzyme and lead to increased trimethylation of the histone X3 lysine transaphane, leading to the suppression of many of the target genes, including tumor suppressors. So it has been known that enzymes can be found mutated in different cancers. But what about the histone genes themselves? Histone proteins are encoded by different histone genes. What if the histone genes are also mutated and targeted in different cancers? So the answer is yes, back to early 2012, at that time when there were two very high-profile papers published. So they were the first stories to report the cancer-driving histone mutation, which is the histone X3 lysine transaphane to methionine substitution mutation found in pediatric brain cancer patients. So what is this mutation? So this is a lysine transaphane, K transaphane to methionine mutation found in the pediatric brain cancer. Lysine can be modified by methylation, acetylation, ubiquitination, and so on. But the methionine has not been shown that can be modified by any of the enzymes. So how does this lysine to methionine substitution affect gene expression and subsequently cancer development? So at that time, when I was postdoc at the Mayo Clinic, we collaborate with some clinicians and we received some patient samples. And the first experiment we did was to do a Western to show that the histone X3, lysine transaphane di and trimethylation were dramatically reduced in the patient samples compared to the control, either with a neural stem cell control or the brain cancer sample without the mutation. So you can see the di and trimethylation of the histone 3 lysine transaphane is dramatically reduced. But then the enzymes responsible for the methylation, the EZH2 and the PRC2 component substrate, they were there, suggesting that it could be affecting the enzymatic activity of the PRC2, EZH2 instead of a downregulation of the enzymes. So we further performed some experiments, including the chip sequencing, and you can see that the histone X3 lysine for methylation is not affected in the patient sample compared to the control, while the histone X3 lysine transaphane methylation is dramatically reduced compared to the neural stem cell control, proving that indeed the mutation affects the histone X3 methylation.
What particular cancer?
Pediatric brain cancer, the diffuse intrinsic pontine glioma, and also some of the
from same patients or from
right from the same patient.
What normal patient without mutating, what happens?
Normal patient without mutation doesn't affect the di and trimethylation. So the mutation affects the histone X3, and the histone X3 act as an inhibitor, inhibit the enzymes responsible for the methylation. So the enzyme can no longer methylate the wild type histone in a cell, in the nucleus. You're welcome. So instead of continue working on the histone X3 lysine transaphane to M mutation, after moving back to Hong Kong to start my own lab, I started to search for other histone mutations instead of the histone 3 lysine transaphane to M mutation. So in collaboration with Dr. Wang Xing at the CUHK, we started by analyzing the TCGA database. So from the TCGA database, we analyze all the patient samples when we also find out all the histone mutations in the histone X1, histone X2A, 2B, histone X3, histone X4. So we did a lot of the analysis. And you can focus on these PDAC, which is a pancreatic ductal adenocarcinoma. So very interestingly, if we look at these 146 patient samples, you can see that there are around 10% of the patients they have the mutation in a gene called H2B1, one of the H2B genes that code for H2B proteins. So we focus on the PDAC, and we found that among that 146 patients, there are 6.9, around 7% of them, they harbor the same mutation, which is glycine 53 to aspartic acid mutation. This mutation can also be found in the glioblastoma and also the lung cancers, which in the glioblastoma, this mutation has been reported by another group early in 2014. So what is this mutation? We first look at the histone H2B, and then we compare the amino acid sequence, and then we found that the glycine at position 53 is highly conserved from E to C. elegans to Drosophila to mouse to human, suggesting that the glycine 53 of the histone H2B may be very important for the normal histone H2B function. If you go back to this human nucleosome structure, crystal structure, you can see that the glycine 53 is located here, which is very close to the double-strand DNA. So the glycine 53 is located in close proximity to the DNA, which is near the DNA entry and exit site. We know that the glycine is neutral, but then the aspartic acid is negatively charged. So the mutation changed the glycine to aspartic acid in this region. And we all know that the double-strand DNA is negatively charged. So the glycine to aspartic acid mutation at position 53 of the histone H2B may affect the nucleosome stability because of the negatively charged double-strand DNA, which is very close to the negatively charged aspartic acid after the mutation. So in collaboration with Toyotaka at Hong Kong UST, we first performed this experiment, which is called the single molecule nucleosome pooling and holding assay, which they are able to perform this experiment at the single molecule level to measure the force required to separate or denature a nucleosome. Measure the force that is able to denature a nucleosome. So we have the wild type nucleosome, and we also have the H2B glycine 53D mutant nucleosome. So they are able to measure the force required to denature the nucleosome. And I would like to let you know that the result is much less force is required to denature the histone H2B glycine 53D mutant nucleosome compared to the wild type, suggesting that indeed the H2B G53D mutations weakens the DNA histone interaction because less force is required to denature the nucleosome. So the weakening of the nucleosome stability might affect many biological activities on the chromatin, including the DNA damage repair, DNA replication, and the transcription. So to address these questions, so we used the CRISPR-Cas9 approach to knock in these mutations in a pancreatic cancer cell line, S2-10. So we put a flag tag at the N terminal of the wild type or the G53D mutant DNA, and then knock into one of the H2B gene loci. And then we pick single colony, and then we perform a Sanger sequencing to confirm that the mutation has been knocked into the cell line. And then we have also tested many of these potential off-target sites, and we didn't find any of the mutation in these 4 cell lines that we have validated. So using these 4 CRISPR-Cas9 knock-in cell line, 2 wild type and the 2 mutant cell line, we test whether the mutation affects DNA damage repair. So we treated the wild type cells and the mutant cells with the MMS, which can and CBT, which can induce DNA damage, and then we perform these 53BP1 or the gamma H2X immunofluorescence staining, and then we count the number of cells, but then we didn't see any difference between the wild type and the mutant after the treatment. We then also collect some lysates and perform the Western blot, and we also didn't see differences between the phosphorylation of the gamma H2X between the wild type.
Maybe your DNA repair not damage not strong enough.
Yes, possible, because we only use these 2 different damage drugs, but then we are currently testing other drugs to show.
Does the mutation affect cell survival?
No.
Cells proliferating?
No, I will show you later. It's not affecting cell proliferation.
I mean, you need a chance to DNA damage stuff.
Right, right, sure. We are currently testing other DNA damage drugs, but at that time we only focus on using testing with these two commonly used DNA damage agents. But anyway, we also test the cell proliferation, and then we also did the incorporation of BRDU into the cells, and we didn't see much differences, suggesting that perhaps it doesn't affect DNA damage repair, and it doesn't affect replication. So we focus on the transcription. The first experiment we did was to do these RNA-seq, and then we find hundreds of differentially expressed genes compared to wild type and the mutant cells. And if we perform these pathway analysis, we find that many of these cancer pathways are affected, including these pathway in cancers, focal adhesion, PI3K signaling, and also cell adhesion and cell movement, so on. So we would like to know whether the effect of the mutation on the gene expression is direct or indirect. So to address this question, we used a flag to perform a cut and run, which is very similar to the chip sequencing. We want to map the genomic localization of the wild type and the mutant G53D nucleosomes. So if you look at these genome viewer of the flag cut and run sequencing and also H2A sequencing, which is a control, you can see that the G53D mutant H2B is enriched in some regions in the genome. And if we compare the cut and run peaks to the control peaks, you can see that many of these G53D H2B peaks is located in the promoter region compared to the chaperone peak, which is the control. And many of these G53D mutant peaks are co-localized with the ataxic sequencing peaks, suggesting that they are located at the open chromatin region. So we then perform these gene set enrichment analysis, and then we show that there's a positive correlation between the G53D enrichment in the genome and also the gene transcription, because we correlate the cut and run with the RNA-seq. And then we find many of the genes are involved. And one of the genes that we would like to focus on, and I would like to tell you, is the so-called ANXA3. ANXA3, annexin A3 is involved in the membrane dynamics and the metastasis in other cancers, such as in the lung cancers, ovarian, and the liver cancers. But then the role of the ANXA3 in the pancreatic cancer is not known. So we see that this is the ANXA3. We find the H2B G53D H2B is enriched at the ANXA3 locus. And then we also perform qPCR to confirm that there's enrichment of the mutant H2B at this locus. We have the RNA-seq, we have the pro-seq to show the transcription is increased in the mutant cells of the ANXA3 gene, and then we have the Western blot showing the expression of the ANXA3 is increased. So how is it able to directly affect the ANXA3 transcription? So then we performed this experiment, which we first treated the cells with the DRB, which can inhibit RNA polymerase II activity. And then we release the cells in the fresh medium and the transcription can resume. And then we collect the total RNA. We perform the RT-PCR to measure the amount of the pre-messenger RNA. And then we can assay the rate of transcription of the target genes, including the ANXA3. So if you look at here, we have these ANXA3. We have the PCR primers targeting the different exon-intron boundaries along the gene. And then you can see that if we, after the release into the fresh medium, the amount of the ANXA3 pre-messenger RNA in the mutant cell is much more than the wild-type cell. At the exon 2, intron 2, exon 7, intron 7, or other intron-exon boundaries, suggesting that indeed the transcription of the ANXA3, the transcription elongation rate of the ANXA3 is indeed higher in the mutant than the wild type. Another gene, SNAP47, which has a similar size, but is not targeted by the H2B G53D mutant, has a similar transcription rate in the wild type and in the mutant, suggesting that this effect is specific in the mutant cell of the increased expression of the ANXA3. We want to further confirm that indeed the transcription is higher in the mutant cell. So we perform this in vitro transcription elongation assay. Again, in collaboration with Toyotaka, they perform this in vitro transcription elongation assay using purified wild type or the mutant H2B nucleosome. And they also have this human polymerase II to do this transcription elongation assay in vitro. So we can see that the polymerase II is more efficient to transcribe through the H2B G53D nucleosome than the wild type nucleosome, suggesting that indeed the G53D mutation affects the nucleosome stability and also affect the transcription rate. So what about any link between the increased ANXA3 and the pancreatic cancer development? So we perform many of these different cell assays, including the cell proliferation assay. We didn't see much significant difference compared to wild type with the mutant cells. But then if we perform this transwell migration assay, we observe a much higher transwell migration of the mutant cells compared to the wild type cell. If we knock down cell migration assay. If we knock down the ANXA3 using the S-sector RNA, you can see that the migration ability is significantly reduced in both wild type and the mutant. So this is the knockdown of the ANXA3 validated by the RT-PCR and also the Western blot. So knockdown of the ANXA3 significantly reduced the cell migration ability. We further perform this gap closure assay, which is also called the scratch assay. You can see in the normal situation, in the non-targeting control, mutant cells that are they're able to migrate to the gap much faster than the wild type cells. You can see this is a 0-hour control. This is after 16 hours, the cell migrate from the peripheral to the gap in the middle. If we knock down the ANXA3 using S-sector RNA, you can see that the migration ability of the cells is significantly reduced after the knockdown of the ANXA3 using these two different S-sector RNA. But then you can still see perhaps the mutant cells may also have a little bit higher migration ability compared to the wild type cells, suggesting that ANXA3 and possibly other genes contribute to the cell migration phenotypes of the H2B G53D mutant cells. So at the end, we would like to show some clinical relevance of the ANXA3 overexpression.
You would go for a little bit see mice data with the mouse pancreatic metastasis.
We have the knock-in mouse.
You have knock-in mouse.
We have the knock-in mouse, and we are crossing the knock-in mouse with the PDX model. We don't have the result yet. Oh. We have the knock-in. We have the knock-in mouse. We have knock-in the mouse. We are crossing with the PDX, KRAS G12D.
Yeah, KPC.
Right, KPC. Right.
Your data, that would be great.
Right. We have published these in vitro studies, and we are currently doing the in vivo, and also doing the CRISPR-Cas9 drug screening.
In vitro migration.
Yes. Right. That's why we have the knock-in mice. We generated the knock-in mice several years back, but then because of the COVID, the mice cannot be immigrated from the mainland China to Hong Kong, so we postponed the experiment. But then we don't want to wait.
And the phenotype one put these genes back to mice alone?
No.
No phenotype.
There shouldn't be any phenotype, because if you think about this, shouldn't be a very strong oncogene. Even oncogene as strong as the KRAS G12D won't create tumor.
This molecule important for DNA repair. If something is DNA repair,
but then based
some kind of UV radiation, gamma radiation could get phenotype. Right.
But then based on the results we generated using the MMS and the CPT, we didn't see. So we didn't claim that it is affecting the DNA damage repair.
But my claim is that it doesn't use good stuff.
Exactly. So I'm not saying that it is affecting damage repair. It could affect, but then we don't have the evidence.
There's no one that KPC much.
Well, exactly. So we are proposing using other. The student has graduated, so we are recruiting other PhD students to continue working on this project. So we have many experiments ongoing. We can collaborate. Right. We can collaborate. we can discuss after my talk during the tea break. Go back to the TCG database, that we are able to show that high expression of the ANXA3 leads to poor prognosis in the pancreatic cancer. And we also show some correlation between the presence of the mutation with high expression of the ANXA3 in the PDAC, and also with the limited number of the patient, 146 patient of the PDAC available in the TCG database, we are able to find the correlation between the presence of the H2B G53D mutation with the poor survival in the disease. Not statistically significant, perhaps because of the low number of the patient sample available in the TCG database. So we propose a model. So the mutation weakens the DNA histone interaction, which enhances the expression of the G53 H2B target genes, such as ANXA3 and possibly others. So this confers additional oncogenic properties in the PDAC. So high expression of ANXA3, which is with consequence of the increased transcription of the gene in the mutant cells, will lead to some other oncogenic properties in the PDAC. So I have shown you our work in the G53D mutation. And actually at that time, we also identified other mutations in the histone H2B, including these H2B E76K mutation found in the breast cancer, and also H2B E113K mutation found in the lung cancers. I don't have time to show you our data on these two stories, but then I'm very happy to have a potential collaboration with one of the audience in this meeting in Japan. So I would like to thank my friends and collaborators in Hong Kong, in the States, and also in the mainland China, especially Wang Xing, who did the bioinformatics analysis for us, and Toyo Taka did this nucleosome stability in vitro transcription assay. We have collaborators in the States providing some of these cell lines and patient samples, and also Chen Ming did the structural analysis, and Joannis provided reagents for the PROSEQ, and to map the active polymerase II. So I would like to thank the funding agencies. And before I stop, I would like to make an advertisement as a guest editor. We are collecting manuscripts for the title, The Role of Histone Modifications, Variants, and Mutation in Gene Regulation and Human Disease. So if you are working in this field and if you are interested in publishing a review paper in this journal, please feel free to submit your manuscript to us. So thank you very much for your attention. I'm happy to take questions.